Hi everyone. So we are going to start post-midterm and get right into DNA replication, and that's going to lead us into mutations because when DNA replication goes wrong, that's usually what we're going to be looking at is some kind of damage in the DNA that is going to break that code. It's going to change the protein code of anything that's coding for, or perhaps if you're in a non-coding region, you can even mutate DNA and like lose promoter sites, enhancer sites, silencer sites, and you can ruin gene dosage and things like that. So keep some of these in mind too. This was a good, interesting question posed by your book, is that when you're starting replication, why doesn't it just restart replicating all the time? Even during the process of replication. And then the last one, as it pertains to what we're doing right now, a lot of antiviral drugs have to target viruses, and the best way that they do that is targeting viral material. Problem is that viruses are basically pure RNA or DNA, so we have to be very careful how we inhibit those. So we'll keep these questions in mind as we go, and we may address, I think, at least these two directly in this lecture. This one maybe just kind of tangentially. All right, so we will start here at eukaryotic division. So the way that this will go, and the way that most ways that this is taught, is that we usually teach you the prokaryotic division mechanisms and then showcase the eukaryotic division. That's usually because prokaryotic division is so much simpler. And remember, that's just in a big circular chromosome. So your book has a very good look into that. It's much easier. And we'll see why. But for the most part, you still have this basic mechanism that's going to happen. So we will start a little bit right here with a eukaryotic direction, but we'll obviously get into both. So in this case, it can go both ways, as you can tell. See how this is going on your leading strand. Your DNA polymerase can go from 5' to 3' just the entire way, and it's never going to stop. In this direction, though, you have to add little 5' to 3' Okazaki fragments in this direction, since you are going the opposite way. So it is bidirectional. One section is discontinuous, though, and it's going to be a lot slower. The other thing is that you have 4.7 billion nucleotides per genome per cell, and these are going to replicate somewhere in like 100 million cells are going to replicate in your body a day, let's say. I think it's even way more than that. So to replicate an entire genome, you have to start at multiple origins that are ultimately going to meet, for example, in a place like this, and they are going to be able to just, kind of meet and become a single new replicated strand. Here you can tell that you have the double replicated strands there, and that's good. Okay, so that's how you can facilitate a very quick replication, is that it's going to be going in both directions. It's going to have multiple origin sites. We'll actually see that these origin sites are oftentimes specific DNA codes that make sure that an origin starts there. That's especially true in prokaryotes and especially true in the plasmids that we use them for. Okay, here's the basic idea of DNA replication. When you have an incoming DNA NTP, you have one that has all these phosphate groups. This 3' OH, and always remember how reactive these are. They are going to attack right here. These electrons are going to move here, and that's going to bump off these two phosphates right here, and that's just going to go off into the cell. And at that moment, then you are going to be able to form up a bond between these right here, and this nucleotide, this A right here, is then going to form a hydrogen bond and fit right in with that T. So as you can see here, here's our finished bond from the reaction up here. This is the basic overlay right here of how you are going to just insert a nucleotide. And then after you insert the nucleotide, the polymerase complex, this big yellow thing right here, once it feels that this nucleotide is now set up and it's good, that causes a conformational change that leads to the translocation to the next one. Perfect. So each time a polymerase is reading, it is going to read if the nucleotides have fit together nicely, and then it's going to shift its protein 3D shape and allow for a translocation to happen right here. That's going to move on to the next one. Okay, so pol is the short name for polymerase. That is going to come up pretty much all the time. You will see pol as that. Polymerases are large complexes. As you can see, this yellow thing has a lot of different sites to it. Now, sometimes these are different protein components that form a large quaternary structure. Other times it's the same protein. So your book will have a lot of those details, and we'll go over some of the tables on this lecture. But for the most part, what I want you to mainly know is that these complexes they're multifunctional. So that's what we're going to go over here. So Nick repair is essentially a function where, remember how we are going to get to, we're going to link up with strands, right? So when let's take a look at this section right here. When this Okazaki fragment is going in here, it's going to hit this purple, and remember that's an RNA primer, that purple right there. So what Nick repair is that when the DNA polymerase actually encounters and it completes its strand, see this is 5' to 3' right here, see that? This is going to get to some RNA that needs to get removed. So you're at the polymerase domain is what's taking care of everything right now. But what happens is that the pol moves a little bit and it starts to encounter this RNA, and you actually disassociate the polymerase replication machinery and you actually engage this exonuclease domain. So remember, these are big, large complexes. They essentially have different active sites for different jobs. And those active sites for those different jobs are only going to become active should they encounter, their substrate. And in this case, your DNA exonuclease right here, see how this is going to engage once it reads that nick that it has to repair, or once it reads that RNA primer. That exonuclease, which is essentially something that chews up nucleotides, is going to engage, remove this, and then it is going to re-engage the polymerase, and it's going to repair this nick. So we've talked a little bit about, or at least it's going to add on as much as it can right there. I know I'm getting busy right here. It's going to keep adding the nucleotides where this was. And then this little nick right here, some polymerases can fix that on their own, but normally what you want right there is actually DNA ligase. Oh, that's so small, no. So that's what's usually going to fix this right here, that tiny nick right there. And we've covered that reaction before. Okay, this is what we've been talking about, is that when your polymerases, so these are still the same megacomplexes, they have their own editing mechanisms. So normally you have this active site for the polymerase like we saw up there. If everything fits, this is a perfectly shaped protein active site for these nucleotides. If everything fits and it's appearing in a pyrimidine pair, everything's fine. There's no dissociation, there's no pressure, nothing. If you have an incorrect base pair, a lot of the time, that pressure is going to exude itself on the sides of your protein active site of your polymerase. This is going to cause a conformational change, and that is going to trigger the polymerase to stop adding nucleotides, and it's going to instead back up, chew off about 100 nucleotides in the direction that it just went because it made this mistake, and then it's going to restart. So it's basically going to lay those tracks one more time. So if it encounters that bulge, like we'd see right here with this C and T right here, it will immediately disengage, and notice how it's shifted shape here. See how the little yellow macaroni protein right here has shifted shape? What that's doing is it's shifting to get its exonuclease active. It's shifting the position of what active site it has on there, and it's going to go use this exonuclease instead. Because remember, that's very different than the polymerase itself that's actually replicating, the replicase. So it'll use the exonuclease to basically chew up a bunch of nucleotides on this strand, and then it's going to reposition after a few of those, and then it's going to go back to repairing. So this is good. This allows you to repair almost, I think, only 1 out of every 1,000 misaligned base pairs like this C and T are actually going to get away with it, basically. That's how accurate your polymerase is. Now, it doesn't always catch everything, and so a lot of times mistakes are simply, luck of the draw, to be honest. You are going to have DNA mutate, and it's not going to repair. Okay, so remember how we actually just covered this on the test, hooray. Remember how DNA polymerase itself, it's actually relying not only on its amino acids here, but those are engaging magnesiums, right? The magnesiums are the one that are going to feel the effect of a poorly paired nucleotide. So see here you have a C and an A. The reason that you are going to engage this exonuclease is that your magnesiums are now no longer connected to your aspartic acids. Okay? So in that case, once they're disengaged, that's when the exonuclease takes over, because there's a conformational change that allows this active site to come in, and what that's going to do is say, nope, we're going to chew this up right here, and we're going to restart. So these little molecules, these little ions right here, play a massive role in making sure that your DNA is doing well. Okay, so again, your book has a great look at how this protein actually looks. It is amazing that it basically is this massive, like, orchestrated subunit to make sure that DNA is being added in this helix right here. Because this is, I mean, obviously everything that we're looking at up here is so simplified to really this beautiful mechanism that's going to happen here. And it is going to completely weave and replicate this DNA, and, you know, very accurately too. This is how complex the entire complex, can actually get eventually. It's basically that you have, it's kind of this POM model that you can look up more details in your book, but essentially forming up this large protein complex is going to allow the DNA to fold through, get replicated, and also detect damage. So, a very important point of replication is the clamp. There are eukaryotic versions of this. So beta clamp is the prokaryotic version. What a clamp does is it is kind of the docking site for the big polymerase cores. The polymerase cores have kind of a hard time finding their way to DNA, and they kind of have a hard time going really quickly. What the clamp does is twofold. It ensures speed and it ensures efficiency by always catching the DNA polymerase. And that's because it can stay attached essentially to this clamp and just basically keep moving, you know, keep on swimming, keep on swimming. That's kind of what the clamp makes it do here. And so that's why clamps are so important, and that's why when you lose a clamp, you basically, you know, DNA polymerization is so bad, you start having mutations, DNA can't replicate fast enough, and the cells die. So sometimes when you have clamp deficiencies, you have a very systematic issue because all of your DNA is going to get damaged. All right, so that kind of ends the basic overview of DNA replication. Like I said, I wanted to do a little less is more here. So we took a look at clamps. Clamps make sure that the polymerase stays on and keeps going the whole way. So we've talked about helicase before, too. Remember how this is going to go unwind in this crazy, like, cool little circle here. Look at that terrible arrow. And it's going to basically unwind the DNA as it goes. Now, downstream of that unwinding, you need one of your topoisomerases. You need to be sure that this DNA is staying underwound and make sure that the DNA essentially doesn't break back here. Because as you unwind right here, oh man, I'm getting bad at these arrows. As you unwind right here with helicase, never mind. God. You are going to cause a lot of torsion right here, and that's, remember, what the topoisomerases are there for. They are going to relieve that torsion. Okay, next big thing. DNA polymerase can't start without a primer. You need to have an RNA primer there. See that little purple right there? It's going to lay down basically a starter track for DNA to start with, and polymerase will bind to that RNA primer and start going. So we talked about ligase a little bit, because ligase is the kind of the final step, is these little nicks left between the Okazaki fragments are the only thing that most DNA polymerases can't handle, and so ligases come and seal those. These are the DNA binding proteins, these little SSBs. Remember how they just bind up DNA? So during DNA replication, you have a lot of exposed DNA and a lot of single-stranded DNA that is still in the process of being replicated. This is super duper scary for the DNA because it's completely exposed. It's not around the nucleosomes or the histones, remember. So you need these little partner proteins to bind it up and basically just keep it safe. They're kind of a little transition molecule that keeps it safe from the outside world while you're still replicating. So all of these are going to go in and be, they're all very important. One of the coolest aspects about the RNA primer, nobody truly knows why an RNA primer is laid and not just a little DNA primer. Nobody actually has that answer all figured out. The current hypothesis is that there are simply more RNA nucleotides out floating in the nucleus, for the machinery to grab, and that RNA with that extra OH group is simply more reactive. So this synthesis as an RNA primer is just going to happen a lot better and faster than if it was DNA. So those are the two main hypotheses. That's really interesting, though, so I really like that. All right. So with all that, this is actually what that big fork looks like. So if you have these parental strands, see on this side on the leading strand, you have a pol 3 core. So polymerase 3 in the prokaryote is the main one that's just going to go zip. It's going to lay down some good DNA tracks, 5' to 3'. You have polymerase 3 cores over here on the lagging strand, but notice how they have to be operating here, here, and we're over there already. So essentially, they have to operate a lot more than the one does over here. You can essentially have two polymerases on this side of this large fork that are both having to basically catch up with the single polymerase on this side. So chemically, that's why this complex is so unique, is that the lagging strands have to catch up essentially, and that's why you have to have more machinery on this side. So up here you have your good helicase, that's unwinding this section right here. You have a topoisomerase upstream here that's relieving, that's not shown. You have the nice little single strand DNA binding proteins that are keeping all of this nice while it's waiting to get replicated. You have an RNA primer, and you have primase sitting right here ready to go. And then you have clamps ready to go right here. And you notice the little purple clamps everywhere. Those are helping the polymerases. This clamp has a loader, essentially. This is a really unique structure. And well, this whole thing is unique. And I know I've butchered it all here, but you can pause it before I did that. This clamp loader is what is going to slide these nice, efficient clamps onto the DNA. That's going to attract the polymerases. That's going to cause them to go really fast. So this entire thing is working towards making a very efficient machine. The main issue is obviously, though, this lagging strand is so slow. It's so much slower than the leading strand. So the proposed model of the lagging strand is something called a trombone model. And remember, like we said, 2 pole need the lagging. You only need 1 pole for the leading. So with the lagging strand loop formation, you are going to form this nice loop here. It's going to buttress out and grow, and you're going to add that RNA primer. Then you disassemble the loop once you grow this big loop right here, and then you add a clamp right here, and then it's going to start going. And then you start again. Reform a loop from this parental source right here. Hopefully I wasn't too fast. You can go over that again, but look how this loop is going to grow right here. Grow, grow, grow. Then it's going to disassemble and kind of explode out. Now it's free, and you see your little clamp right there. Now it's free to start adding nucleotides here, and then do it again. Go further upstream this, get more loop growth, and essentially start again up here. So that's the trombone model, essentially that it is going to grow out, explode, polymerize. Grow out, explode, and then polymerize. So hopefully that's good. This is something I'd like to be teaching in real life, but here we are. So another cool thing about clamps is that these big proteins, they get recycled really quickly. You don't have to have new clamps all the time. Once one is finished, it's going to disassociate and basically not only guide the ligase in, but part of that reaction is going to allow it to be targeted by a little targeting mechanism protein right here, and this is going to find its way to the clamp loader. Remember that little alien-looking thing? So after it not only guides the DNA polymerase, it's going to find ligase. After that, the ligase is going to allow it to be tagged by the clamp loader. It's going to find its way to the clamp loader, and it's going to, again, be loaded up, usually on one of these loops right here that we've seen here, because every loop is going to need and make sure you have one. So essentially, this little clamp is going to get reused over and over again at this gigantic fork. Okay. So this answers one of our big questions. Why doesn't DNA just start replicating again? The main thing here, and so this is at an origin, that's what ori usually is going to mean. You'll see this on a lot of DNA plasmids too, is that these are going to get hypermethylated all of a sudden, and these are going to discourage the DNA binding proteins that you see usually initiate the process of DNA replication all over again. So that's how. And so again, methylation not only imprints for parental strands, things like that, it marks all these sites of origin as like, okay, we're good. We don't want to have any more replication. This also does a very good job of simply closing up that DNA and making sure that we do not, we don't open that. So this is, again, kind of a cool hypothesized territory area. To get at some of these origins of replications, people were never actually sure what the trigger was that could open them so well. And it turns out, of all things, it's actually, in a lot of cases, we believe, RNA polymerase, just that normal gene that's going to make RNA. It has a very good way of opening DNA up to remember to get at it and make sure that it can make RNA from these lengths. But a lot of scientists have figured out this actually shows up at a lot of origins and doesn't make any RNA. It's basically there just to split and cause this big bubble to form up so that you can start putting in all this machinery here. So I thought that was super interesting too. So I just wanted to point that out, that sometimes, you know, different proteins are going to have a very unique role outside of their main role sometimes. All right, so here we are at the eukaryotic fork right here. It's a little more complex. There are a few more players at role, but again, you mainly still have a primase, you still have a helicase, you still are using RNA primers, you still have a clamp loading loop, you still have polymerases, you still have a clamp itself, you still have a clamp loader, and a couple other things that are going to, right here, mainly facilitate this all staying together, I would say. That's an oversimplification, but we'll get into that. Okay. So a big thing here is that notice that this is polymerase delta, this is polymerase epsilon. They are related, but they are not perfectly, they are not perfectly symmetrical when it comes to their function. We're going to learn a little bit when we get into mutation why certain regions that mutate pol epsilon are very detrimental, and you lose all the accuracy that you usually have while making this strand, and it can lead to devastating results for a cell or for an organism. We'll kind of take a look at pol delta and how sometimes, although this is probably your most accurate polymerase, I believe, don't maybe check me on that, this one has a lot of fail-safes to make sure that it's still working as well. Oh yeah, and one last thing, we have RPAs, so those are like the little single-stranded binders as well. So again, I want you to notice how similar this is. We basically renamed a lot of things. It is more complex because you do have a lot more things to take care of. This is a good representation of that. So we covered right here. These are our three polymerases, for example. This is a primase, so this lays RNA. These two are replicases, so these are like our typical polymerases. Yep. There's a clamp loader, and then you see that a lot of these are scaffolds, basically. They keep this entire fork basically going. All of them need to be around in a lot of cases to make sure that that fork is good, and this replication complex, this big massive thing, stays. But as you can tell, the function of a lot of these are kind of just jumped into, hey, this is how we load this up, this is how we have to destabilize the helix to make sure that we're replicating right, and so on. Oh yeah, and then of course we have our topoisomerases. And then here you'll kind of see, you know, there are helicase loaders, there are certain things that do more pre-replication stuff. A lot of the times these are going to be, there's a lot more steps to loading up a helicase and like opening origins and stuff. Like this ORC is a really good example of something that has to initiate cell replication. A big thing with eukaryotic cells, remember, is that we have a complex, mitosis, S phase, things like that. So when the cell's ready to divide, it has to go through the S phase. So these cyclins right here are big growth factors, but one of their jobs is to go phosphorylate targets that are going to initiate ORC, for example, and all this cascade. So a lot of the instructions to start cell replication in a eukaryote have to come from cell replication machinery that's like, we're ready to go for mitosis. So it's much more complex, and there are a lot more yeses and nos and checkpoints that you have to get through in a eukaryote to divide than you do a prokaryote. Okay, now this is where we get interesting. So when we get to the ends in eukaryotes, in a prokaryote, it's a circle, and you meet at the end of the circle and everything's fine. When you have lagging strands, though, you have a little part that has a primase, and when you chew that primase off, you can't load any more polymerase on this end right here to make new DNA. That's where you start losing these little bits of DNA right here. That's the end replication problem in eukaryotes. All of you are, between 20, 21, etc. You have 21 years of this happening to the ends of a lot of your chromosomes, okay? Now, not all your cells always directly come from the parent cells themselves. You have stem cells in your body that renew your populations. But again, this is a key factor of aging, which is, we are essentially losing this section right here progressively, and eventually you damage enough cells like that they start to lose function and your organs start to have less function. So look at this like right here. If you keep losing little bits of DNA each time, these are going to keep getting lost. And we've talked a little bit about this in class. You progressively kind of chew at the end of each of your linear chromosomes, and that's the key, is that these are linear chromosomes. They can't complete from the first time. So the answer is something called telomerase. It is a hybrid protein and RNA enzyme. What this is going to do, so see, this is the end of DNA. It is going to, let's say this is the usual end of DNA. It's going to grab that usual end that would get chewed off, and it is going to bind it up to its RNA primers, and it's going to lay a new fake track of DNA on the ends of your chromosomes. So this is perfect. This is going to keep your chromosomes renewed at their ends because it's always going to allow for a space for polymerase to come. So the protein part of telomerase is called TERT. The RNA part of telomerase is called TR. Now, losing either, we'll see that both of these play a role when, this gets out of control. But C here is an end replication problem. We're not going to be able to fill that section right there. So what you can do is essentially, oh, sorry, I actually want to make that bigger. You can load, and see how this length is getting bigger. You basically just load repeats using this RNA as a template. You're going to get new nucleotides to bind right here and basically extend this strand, and that's the key. Once you extend this strand, then you can add a nice RNA primer, and then you can just do the polymerase as usual. And now you're never going to lose the ends of your chromosomes. Now, we knew something like this had to happen because if you think about it, think about the sex chromosomes the sex cells, they have to pass down each generation. You could not have been passing down, DNA for thousands of years or millions of years like we have been and have little pieces shedding off each generation. That wouldn't last very long. That'd be really bad. So we have this enzyme that makes sure that linear chromosomes had a failsafe. And this is a very interesting and important molecule because, as I highlight right here, telomerase probably had to evolve before linear chromosomes. It probably had to have some kind of other role as like a DNA repair mechanism because you couldn't have linear chromosomes without this. If you did, you'd just keep replicating and leaving chunks off of it, basically, and that'd be terrible. Well, the chunk would actually be up in this whole chunk right here, see it? So that's why telomerase is so important. And we at first were like, oh my God, yes, we could cure aging with this. We could stop DNA damage with this. Unfortunately, cancer cells love activating this pathway because cancer cells, when they divide, they lose too many of these chunks right here. See how this whole section would go away? Your chromosomes lengthening is actually a pretty good way for cancer cells to just go away. If any of you have a mole, for example, those are tumor cells that basically just got exhausted. And in a lot of cases, that's because they lost the ability or never had the ability to add more length on their chromosomes, and thus they would lose these little spots right here. When you activate telomerase, unfortunately, it is likely going to lead to a cancer of some kind because your cells become what we call immortal. They will not degrade because they will never lose these chunks, basically. That's why the sex cells that you have are essentially immortal because they will not degrade over time. They're always going to be augmented by telomerase. So that was unfortunate. We're still to this day looking at how to harness telomerase for anti-aging purposes, but unfortunately we're not amazing at it. Okay, so this is something that we'll discuss on Wednesday, and it's something that I usually ask most of my classes, and we've gotten to the kind of an advanced point that I want to ask about. And so I usually ask people what constitutes life. So we have a lot of biochemists in our class, which is good. They can tell you, just like all of us can, you have a living cell when all the components are in a proper order and conformation, but you have non-living things that create this living thing. So although the sum of the parts is not living, the synergistic application and combinations that you can create are living things. So in this case, it's good to kind of assess where do you draw the line at what creates life? What is something that is what qualifies essentially. Usually where people run into issues is viruses, for example, because they're not a cell, they're not harnessing their own or producing their own energy, etc. But even so, I'd kind of want to address this more in a human and healthcare way. So we are eukaryotic organisms, and we have eukaryotic cells, each of which is technically an independent living thing, right? So if you look at this following scenario, if somebody takes off an organ from somebody, like an appendix, let's say, those are your cells. They place it on a surgery table. And in that moment, are those cells living or dead? Physiologically, they're definitely living. They contain your DNA. They're part of you, right? But they have been detached from the larger organism. So philosophically, I want you to kind of think, where in that realm is that still a living thing? Are those cells living even though that now they're kind of detached from their main mothership, let's call it? And so if you say yes, they are a living thing and they represent you, a living person, let's take a look at another scenario, and we'll address this scenario right here. I don't know if I've gotten to my fun morbid example that we'll cover, but okay, these are cell lines. I've talked about these before. These are lymphoma lines right here. These are the cell line names. This is something called cell of origin, so it's the type of lymphoma. This is the age of the donor, the gender, and the status when these cells were extracted. So these were taken from patients, removed, and basically given the right resources and environment to live in a lab. These cells, as long as you take care of them and split their populations over and over so they don't become overcrowded, will live forever. If you feed them serum and, kind of the glucose and vitamin solution that we feed our cells, the media, they'll live forever. Now, each one of these contains the full genome of the patient that they came from. If you answered up here that person, let's say you take this out and put it on a tray, that person is on that tray, let's say, then it's equally haunting to imagine, are these people still alive or not? Their DNA exists, their cells still exist. Technically, they're not going to go away, right? So then this question becomes the one to address. How many parts are lost before an organism's gone? So I would do this in class just on the chalkboard, but essentially if you have a person, oh boy. Okay, there's my little stick figure, hooray. If you have a person and you take off different parts, right? You could take off, arms, legs, lower half, you could even take out somebody's heart and put them on a bypass machine, for example. But if you ever mess with a brain in a human, that's when most people usually draw the line, and it's like, okay, that person is now, we're gone from that person. And even in that case, you didn't actually even change the DNA. So kind of a whole nother question. So as we get into this idea of, immortal cell lines and how long something can live when it does have a supercharged telomerase, and all of these do have a supercharged telomerase in the tumor that they came from. That's why they lasted in the lab, and that's a big reason why. But it's a good thought experiment to kind of go over and think about, what's left of a person, even when it's like this. So I always think about that because this cell line here, U2932, it was probably my primary cell line that I used throughout my thesis as a graduate student, and she died from this disease at my age, and I always kind of think about that whenever I read through my stuff, and I'm just like, I have no idea who this person was, but they still represent, are they still in this, in the world essentially? And it's very, I don't know, it's very hard to reason that. The reason I have all this philosophy is that Henrietta Lacks represents the first patient from which cells were collected that could live in lab. These are called HeLa cells for Henrietta Lacks. We still use HeLa cells to this day. Her contribution, and remember, It was not a willing contribution. She was being treated at Johns Hopkins. They took a sample of her tumor and tried to grow it in the lab, and it worked. Johns Hopkins, to this day, owns those cells. They sell them. They keep them as the pure population. And to me, that's always kind of brings up questions of who and what these are. Are they human anymore? And that's a terrible question, because this was obviously a person, and her contribution, like I said, not willingly, led to medical science as we know it today. I guarantee that. You would not have the same advances in DNA, therapies, cancer, disease, virology, anything, if you don't have human cells to work with. Because we can't just do a lot of the experiments that we do, especially like even what I do, for example, on people. That's terrible. You can't do it on mice either. These represent the only way that we can study stuff. And now we obviously harness that process so we can have thousands of cell lines from different patients that are now willingly, at least, to sign up here. So for me, as we discuss DNA and genetic material and like what role that plays in who someone is, it's a good question to kind of ask yourself. So we'll discuss this a little bit on, I think, on Wednesday. So again, this is a good example of people are still trying to harness telomerase in the right cells at the right time. Again, though, every time we've given or activated telomerase in the regular cells of a mouse, they usually get cancer. If we could harness telomerase a little bit, we could probably find ways to eliminate some of the issues of aging. I would not be surprised if this is possible. I would be more surprised if we can harness it safely within the next 50 to 100 years, let's say. Okay. All right. So now we're going to start our intro into DNA mutagenesis. This isn't from the next chapter, but it's going to kind of talk about the primary articles that I'm going to have us read a little bit on and take a look at mutagenesis. Most mutagenesis issues, are going to lead to human cancers. That's usually what happens here. So it's very good to kind of know their roles and exactly what happens when you lose one. So here's our two polymerases, pol delta, pol epsilon. You have some others. So see, pol gamma is actually a mitochondrial DNA repair one, which is really interesting. So some of these, and we'll get to know all of these different repair mechanisms as well. Some of these polymerases are purely there for, different repair mechanisms. In a lot of cases, you need to repair, or you need to facilitate breaking and re-breaking, or unbreaking and fusing DNA, for example, like when you're crossing over. So some of these are only active in certain times. So pol epsilon and pol delta are usually the ones that show up. Now, if you lose one of these main ones, one of these less accurate ones is going to take over for you. That's when you have a big systemic challenge of, we are going to have a lot of mutations here. So that's bad. Okay. So some of the articles that we'll cover will kind of showcase the issues that can arise when you have a non-functional pol delta or pol epsilon, because these are the two that are really going to matter for that nice, accurate polymerization. Okay, so this is a very good example of this, and so like I said, you're going to read some primary articles, but I'm not going to make you analyze them. I mainly just want the main points to come across, and this is one of the main points that I'll ask you. And so when you have a wild type pol epsilon, once in a while you're going to make a mistake, but you have a nice exonuclease domain. Remember, this is going to chew the bad DNA up. It's going to get it right up in here, chew it, and then it's going to relay that primer. Okay, so if you completely ruin pol epsilon, a lot of the times you'll get another polymerase that will show up. It's not great because a lot of the times they don't have an exonuclease domain that's very good. So they may have to rely on different repair mechanisms, which are never as accurate as your initial ones. The fork may collapse, which that's bad, but it also can get fixed. So in this case, if you fully delete pol epsilon, you end up with a few mutations. It's not bad, but it's definitely not great. Here's where things get interesting, and what people were so puzzled by was that when we could finally start sequencing DNA, we found the most damaging mutations were always if you had a point mutation that just knocked out the exonuclease activity. Without it, pol epsilon's still going to go on as though it's fine, but its exonuclease is just going to keep leaving tons of mistakes. So if you mutate the exact right amino acid in the exonuclease, the whole thing's bad, and it doesn't get replaced by something that like, okay, it's not a perfect substitute, but it's okay. It just keeps going and keeps making mistakes because it doesn't know that this is ruined at this point. This is where you get into full issues. Okay. All right, last slide. Oh no, we can't see it. Oh no. Here, I'll do that. There we go. Never mind. Come on. Okay. Well, shoot, I can't get it to here. I knew I should have had another slide. With the Ames test, what we're going to do is take a Salmonella strain that cannot live without histidine, and we're going to plate it on a plate of histidine. So we're going to take this bacteria, we're going to put it on a non-histidine plate, and we're going to see what happens. One, in a condition where we're going to add a suspected mutagen. So this is where I'm going to ask you guys to contribute, because I'm going to do this experiment and then give you the results to write about. Then we're going to put it on the histidine plate, and voila, if you have a bunch of revertants, that suggests that this mutagen actually did cause these to all basically mutate the histidine pathway so that they can survive without histidine somehow. Then you have a normal plate over here that may have a couple revertants, but if it's not a mutagen, it's going to be about the same. If this is a mutagen, you're going to have a ton of survivors that like basically mutated their way into survival with the help of this thing right here. Okay, that should be good enough for today. We'll cover chapter 12 more explicitly once probably maybe later this week, maybe next week, we'll see. I do like exploring this stuff more on a like disease standpoint than just going over everything and being like, you know, this is this repair mechanism, this is this. So I may just have you choose one to kind of talk about or post about, and we'll post like a large collection as a class. So that'll be good. And then we'll prepare for this lab next week, the week of the 17th. Is that it? I don't know. I think that's the Friday. We'll see. All right.